And good morning, everyone. It is Friday, although it doesn't really feel like Friday, and we're going to be continuing our discussions. Been some questions about different versions of the text and what you should be studying. I'm going to work on trying to figure out to make sure that I know how the 11th edition compares with the other ones we're using. The 7th edition is pretty easy. It is, I think, nearly identical to the 8th. On the syllabus, I think it said chapter 22. It should be, I'm pretty sure, chapter 2 in the 9th edition. We're looking at chapter 1 and chapter 2, which is basic intro stuff. And as we continue, I'm pretty sure that all of the volumes, or the editions, the first 5 or 6 chapters are identical. And as I've got the 8th. I'll look to see what the other editions look like. So we've been talking a little bit about basics of microbiology and some of the basic discoveries that led to the field that we are studying, the field of microbiology. First people had to recognize that there were, in fact, microbes. And second, we have to then begin understanding what kind of organisms these were and how they actually began to work. And we'll be talking a little bit more about the classification later. Guys like Pasteur were understanding how sanitation could work and sterilization could work. They began actually to experiment with vaccines, the ability of, or the observation that somebody that had been exposed to a pathogen once wasn't going to be sensitive to it a second time. And we're starting to see that actually with the COVID virus. Something I think it's important to note is that while there have been some discussions of people getting reinfected, those folks that are getting reinfected are either asymptomatic or have dramatically reduced very mild symptoms. So your immune system does work. All right, another thing that was brought up in chapter 1 was the discovery of antibiotics. We'll be talking more about antibiotics as we get into this. Alexander Fleming discovered penicillin, which was a product from a mold, Penicillium. The mold secreted a material that, actually, as it turned out, interferes with the construction of the bacterial cell wall, and that is now known, of course, as penicillin. And this was a very major breakthrough because prior to the discovery of antibiotics, bacterial diseases could be very dangerous. And one of the treatments would be just simple amputation of an infected part of the body because there was no way to treat the bacterial infection necessarily, and it could spread. I'm standing in the Leidy Tabor Science Building, and Dr. John Leidy worked at Abbott Labs? No, not Abbott. He worked at a lab at a company in Indiana, and he was involved in developing some of the systems that could be used to stabilize penicillin, because apparently penicillin that you extract from the mold isn't very stable. And so John Leidy, who this building is named after, he actually was part of the team that was developing some of those technologies back in the 30s, or no, the 20s probably. All right, so let's see. The last bit in chapter 1 gets into some of the ideas of molecular genetics, which is one of my specialties. The idea that bacteria contain DNA and RNA. They actually use very similar mechanisms to go from DNA to produce a protein, and we'll be talking about the flow of information from DNA to protein later in the semester. And that has been used as a tool to manipulate bacteria, but also as an understanding of how bacteria can transmit information among each other. We talk about, you've heard about the increase in antibiotic resistance in bacteria. Antibiotic resistance turns out to be transfer of DNA from one bacterium to another. And that can be between two different species even, or individuals. And so the the phenomenon of antibiotic resistance is genetic. What makes a bacterium resistant to an antibiotic is it now has a gene that is able to either detoxify the antibiotic or a gene that produces a protein that is not being interfered with by the antibiotic. And so that's how antibiotic resistance can spread. It spreads through DNA. So we're going to move into chapter 2. Oh, and one other thought. I'm going to be posting some study guide type stuff and some PowerPoints that you can go through and sort of supplement the lecture with, and we'll see how that goes, all right? So we're going to get into chapter 2 now, which is basic chemistry stuff. And this assumes that you've had basic chemistry, that you've had essentials of chemistry. But we're going to go through it a little bit and talk about chemistry sort of in the way a microbiologist might think about chemistry and the critical parts we think about for the concepts that we'll be talking about. So first off, let's start talking about the fundamentals, and that is the atom. So the atom is the smallest sort of individual subunit of difference in matter. You get into subatomic particles, and the subatomic particles are common to all atoms, but atoms themselves can have specific characteristics. And so we start talking about the idea of the element. And you've seen the periodic table. This is the list of elements. And what this is a systematic way of organizing the atomic structure of every element. So the atom, we're going to start first with the atomic nucleus. Now, in eukaryotic cells that have nuclei, like you and I, and plants, but not bacteria, the nucleus is the site where the genetic material, where chromosomes are. But in the atom, the nucleus is the site where we've got two subatomic particles residing. One being a proton, which is carrying a positive charge, and another subatomic particle that we call a neutron. And it's got no charge. It's free. Now, these two particles are at the core of the atom, and then surrounding this is going to be another charged particle called the electron. So the electron is a negatively charged particle, and it is zooming around the atomic nucleus. So just one of the things I think is interesting to think about for scale is that imagine that you're on a football field, and this football field is in a domed stadium. So you got, really way up there is the top of the dome, and then the dome stadium actually continues below you, so you're really standing in a sphere, in the middle of a sphere. Now, take a basketball, boom, 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 and put a plus sign on it, put that on the 50-yard line, that's a proton. Take another basketball, put that next to it, that's a neutron. So that's the kind of relative size we're talking. Now, an electron is a good bit smaller. Electrons are going to be the size of, a tennis ball, between a tennis ball and a ping pong ball. And you're going to take that and throw that up into the ceiling, and it's zooming around. And really, there is no ceiling. The ceiling, the sphere, is the track that the electron is zooming around. Now, the elements are dependent on the number of protons that are present on that 50-yard line. So if we look at our drawing here, with one proton, one neutron, we have diagrammed hydrogen. This is the atomic nucleus of hydrogen. Now, if we add another proton, right here, instead of hydrogen, we've now made helium. So, one of the truisms, and an important point, is that an element is defined by the number of protons that it's got in its nucleus. The neutrons actually are irrelevant. You can have proton number changing, that changes the element identity, but the neutron number doesn't change, and it's still this different elements. So one of the terms we use, and this is a little bit of a simplification, but it's worth it for now. We have this vocabulary word that we call an isotope. An isotope is an atomic nucleus. Can you see that? Isotope, I-S-O-T-O-P-E. It's an atomic nucleus such that the number of protons doesn't equal the number of neutrons. So one thing that's important to recognize as well is that this is a really good working definition. I like this as a definition. In the chemistry world, they talk about an isotope being, or the element itself, a non-isotope, is the most common form of that element on the planet. And it turns out that with hydrogen, the most common form of hydrogen is a proton and no neutron. And there are forms of hydrogen that don't even have an electron. And we refer to this as either a hydrogen ion or a proton. But for the most part, we can go with this. The number of protons determines the element's identity, and it doesn't have to equal the number of neutrons. Some isotopes are not stable, and they'll kick out subatomic particles. That's radioactivity. And if they kick out neutrons, they are not changing elemental identity. They just are simply changing their weight. But if they kick out a proton, they actually change elemental identity. And with some isotopes, radioactive isotopes, they will have a neutron convert to a proton, and it'll actually change its element identity and go up one spot in the periodic table. Most of the time, they lose a proton and they go down one spot in the periodic table. Just a little bit of trivia. There you go. Thank you. All right, so that's an element. Now, we can begin to link individual atoms. And from here, we start to make molecules. And these definitions, whatever, that's fine. The main point, though, I want you to know is how we begin to link individual atoms. And we do that by creating what are called bonds. And this is, in fact, a critical bit of information here. And that is the idea of chemical bonds. And when we start thinking about chemical bonds, we can categorize them into primarily three categories, and then there's a few hangers on. But there's three bond types that I want you to know and be able to define. And we're going to list them in the order of their strength, going from the strongest to the weakest. Number one bond is something called a covalent bond. And this is defined as the equal sharing of electrons. Now, in order to get a covalent bond to form, we have to get two atoms very close together. And something to think about is the atoms are going to have electrons surrounding them. Those electrons have negative charge, and then we have these two negatively charged particles are trying to interact with each other. You know, opposite charges attract, like charges tend to repel each other. And so what we're going to do is get these two atoms. So here's the subatomic nucleus. Here is the electron of atom 1. Here's the subatomic nucleus. Here is the electron in atom 2. We got to get them close enough so that now the two electrons are playing tag and chasing each other. That's pretty close, and that requires a fair amount of energy. a way to think about bond strength is the amount of energy that it takes to make the bond is equivalent to the amount of energy that it takes to break the bond. Now, happily in biology, the kinds of covalent bonds that we're going to be talking about are typically found in organic molecules that you have pretty good familiarity with. And we can think about what does it take to break these bonds is essentially the amount of energy it takes to set them on fire. Fire, my friends. That's a way to think about how much energy it takes to break a covalent bond. And I'll just tell you from a cooking standpoint, it's around 450 degrees for most organic molecules to begin to break apart, to set them on fire. So if you set your oven below 450, you're not going to get any char. Stuff won't catch on fire. Now, if you set it above 450, you probably better keep an eye on it because it could, in fact, catch on fire. So 450 degrees, that's very hot. And we're going to see how this is going to play a role in some biochemistry because we're going to be breaking bonds a lot. And if we have to generate temperatures that high, there could be some problems. I can't generate 450 degrees in my body. That's Fahrenheit, centigrade, whatever. I can run a fever of about 104 maybe, 105. If you're a little kid, you might be able to pull 105. I probably can't. And that's nowhere close to breaking covalent bonds. But we'll talk about some of this as we get into it. So that's the number one bond. Covalent, strongest. Only way you break it is to set it on fire. All right, our number two bond, and the number two guy is a good bit weaker. The number two bond is called an ionic bond. Now, an ionic bond is made up between two atoms that have a charge to them. That is, actually, we define an ionic bond as unequal sharing of electrons. What does that mean? Well, let's think about a standard molecule that's made up of ionic bonds, and that would be salt or sodium chloride. So we've got a sodium atom, and the sodium atom is missing an electron, which means that it is carrying a net positive charge. So this is the charge on sodium. It really wants an electron. And then here's chlorine. Chlorine is carrying an extra electron. So that means it's carrying a net negative charge. Now, what happens is the sodium really wants an electron, the chlorine really wants to get rid of its extra electron, and bada bing, we end up having a match made in heaven. Now, ionic bonds don't take a lot of energy to make, they also don't take a lot of energy to break. And consequently, if you take salt and put it into water, it'll break apart very quickly. It'll ionize into sodium ions and chlorine ions. Now, this only happens in water. In a dry environment, salt actually is pretty tough to break apart. But biochemistry, which is what we're talking about, is chemistry that occurs in water. So we're safe there. All right. So that's our number two. Number three, I think, is my favorite bond. And number three is an interaction that happens when we have what's called a polar covalent bond. It is typically known in the business as a hydrogen bond. Not to be confused with a hydrogen bomb. A hydrogen bond happens when we've got covalent bonds, which are supposed to be equal sharing, but you know how this can be. Equal is, equal does. there are some atoms that are referred to as being electronegative, oxygen being one of them. It's an electron hog. And so we can look at a good example of a hydrogen bond in a molecule of water. So water, H2O, like this. The electrons tend to hang around the oxygen. Once in a while they get out here, but mostly they're hanging here. And just remember, whenever you see the line is a covalent bond, in these chemical structures. All right, so the oxygen is carrying a slight negative charge, and the hydrogen's carrying a slight positive charge. Now, with a single water molecule, not much happens, but you put water molecules together, and we end up with this charge interaction occurring. We start making a mesh network of the slight positive charge, the slight negative charge, and this is what makes water form great droplets. It's what makes water actually a fairly weird property, because water has some fairly strange properties in that it's very sticky, and that's because of all the hydrogen bonds. So the fourth interaction, which can I put it up here? I'll put it right here. Number 4 is very weak. It is known as van der Waals forces, named after Professor van der Waals. And van der Waals forces are very weak interactions between two atoms that are very, very close to each other but not forming a bond. It's not even an electrostatic interaction, but it's something that actually occurs in large molecules, which we'll be getting into in just a moment, that have a variety of different kinds of great big structures that are next to each other. So that's the basic chemistry we need to know. Let's see. Taking kind of a jump now, going from these individual atoms, let's start linking them together. And what we're going to do is make molecules that we find in living systems, biomolecules, these biopolymers. And one thing that we see in living systems, bacteria, you, your dog, is that the molecules tend to be very, very big. They are what we refer to as macromolecules. Now, macromolecules begin to have some really cool overall structure and interaction because they are so large. And macromolecules are really the stuff of life. And they tend to be, pretty much always, what we call polymers. And a polymer is a macromolecule made up of a bunch of repeating subunits known as monomers. And so a way to study and memorize and learn these guys is to know the monomer and what are the polymers they make up. Or the other way around, is know the polymers and what are the monomers that make them up. That's how we'll do it right now, is we'll put together the monomers and then link them together to make polymers. And we'll work our way through the 4 classes of biomolecules this way. What are the monomers and what are the polymers that they make? Now, just as a general notion, whenever you're building these guys, polymers are built up of these monomers. The monomers are typically linked through what's called a dehydration reaction. This is an example like this. We've got some molecule. We're going to just do this. And what I'm drawing here is one of the base monomers known as an amino acid. So we've got an amine group, 2 carbons, and then on this carbon, so carboxylic acid. You remember this from organic chemistry, I hope? Good. Go back and review. And when we link it together.
Hi. Microbiology.
Yes. Who are you?
Anna.
Hi, Anna. We're in the middle of a microbiology lecture, and you can be the star of it.
No,
Say
it's
hello.
okay.
Look,
Okay.
I'm not sure how to pause this thing.
Just stop it. I'll cut the video later. I have Premiere.